we'll move on to the second speaker. The next speaker is Dr. Morgan Marks from Merck, and he will be sharing with us his work on characterizing burden of dengue from patients' perspective.
Okay, great. Thank you very much. So I want to first thank the organizers for the invitation to present this. What I'm going to do over the next 19 or so or less minutes is just give an overview of a story of the development of a patient-reported outcome tool for measuring dengue fever in a non-hospitalized setting. So this is a story that spans a number of years, and I would say some of the collaborators in the audience would say that the story is epic given the number of years that it's taken and the headwinds and things like that. But so how do I move this forward? So this is my disclosures. So starting with kind of the rationale for how we think about burden of dengue illness and why, a patient-reported outcome tool or measuring the subjective experience of the patient is somewhat important. So I think as it should be, the burden of dengue illness is typically characterized as by the numbers, number of cases, a number of dengue fever cases, number of severe illness cases, number of deaths, and then that data feeds into additional metrics on QALYs and dailies and feeds into modeling data to understand the burden of illness. In addition, when we talk about dengue illness severity or intensity, that's typically characterized and quantified in many ways through the lens of the healthcare provider, which is very important. But there's very few resources that exist that actually try to understand dengue fever from the perspective and the lens of the patient. And that talks about how a patient is experiencing their dengue illness, particularly when they're outside of a clinical setting. They're at home with dengue fever, riding it out, on their own. They're not hospitalized. And also how that dengue illness potentially impacts their quality of life, how they go or not go to their job, how they deal with family things. So characterizing that subjective experience, which is different for everybody, and trying to quantify it in some way may help in some ways in the research setting to add important information, complementary information on how we think about burden of disease, as well as providing a potential additional endpoint that could be used for the evaluation of interventions, therapeutics, vaccines, in terms of reduction of the intensity of dengue. So, getting ahead of myself. So how do we kind of go about this? Well, our starting point, and I said this was a long story, so we're going back to 2016, I think. This is the Dengue Illness Index Report Card, which was designed by Dr. Thomas and other experts in the field, Dr. Edelman and a bunch of others, to really try and first capture from the patient's own experience, kind of their sign and symptom occurrence. So this is a fairly straightforward report card where a patient is given this card after they report dengue fever or dengue symptoms, and they start filling it out on a day-to-day basis. And this is through day 14. They check off whether they have the kind of list of signs and symptoms listed, as well as kind of measure, and then the duration of the symptoms is calculated based on the check boxes. In addition, there's a kind of global measure there of greatest impact where a patient is asked, or a patient takes this card and kind of each day of their illness assesses how their illness affects them on a daily basis. And you can see the Likert scale there with the smiley faces and not so smiley faces. So that was the starting point. This is the end point, or at least the current end point. So I'm kind of giving the spoiler, giving the end first here, which is this is the Dengue Virus Daily Diary, which is a patient-reported outcome measure that was developed starting with the Dengue Illness Index report card and then going through a multi-step development process, which I'll walk you through. But you can see that there are similarities and differences, right? The main similarities are there's a list of symptoms that are developed based on patient input, based on scientific and clinician input, and you can see the language there is a little bit more relatable as a patient, would fill this out at home, experiencing their illness. In addition, you can see that instead of yes, no, the occurrence of those symptoms, you see a scale, right? You see that symptom is non-existent or, it impacts them or affects them a lot. In addition, there are two measures of impact, one on general health and one on productivity that were developed. And then there was a measure related to over-the-counter medication use that in any sort of self-reported measure would be an obvious confounder that we'd want to measure, and then measurement of fever. Another important thing to mention here is that this is a screenshot. So this tool has been developed solely for electronic deployment, either on a provision device, or this was validated on patients' own individual smartphones or their own devices. So it increases the accessibility of a tool like this. So here are the steps that we went through over the years to get to that endpoint. So we first started with the instrument refinement step, where we consulted scientific experts, we consulted guidance documents put forth by the FDA and other professional organizations like ISPOR to really make sure that it follows the guidances that are put out by health authorities. Second was a qualitative interview process, which I'll talk about, where we basically identified patients who had recent dengue illness in Latin America, and we went through a series of semi-structured interviews to identify dengue illness symptoms, as well as understand whether this tool is comprehensible to them. Third, we were presented with an opportunity to quantitatively assess a early draft of the tool in a human challenge model for dengue 3. And then lastly, we went through a formal psychometric validation in a prospective cohort study. So starting with the instrument refinement, and I'll just touch on some of the high points here, just to demonstrate how the DIIRC was then kind of adapted into the Dengue DD, or at least an early draft. You can see that, and I'm sorry, my eyes are not great, but you can see that, so for example, when you look at malaise on the DIIRC, that was translated to, I felt tired. So kind of terminology that for a patient in the field would understand. And then, for example, abdominal pain, my tummy hurt, is another example of kind of that adaptability or that translation. It's important to note here that this is a version of the tool for adults. We also are developing or have developed a version for caregivers of children who experience dengue. Unfortunately, and I'll talk about this as a major limitation here, we weren't able to get the numbers for the psychometric validation. We have the tool, but we're still in the process of validating it right now because we have to accrue a number of cases for that version of the instrument. In addition, when we talk about kind of global measures of impact, so the DIRC had a single measure of impact. Here we broke it up into two. Again, one was for general health in the last 24 hours, and then one kind of tried to measure productivity level of an individual. So for qualitative interviews, and I think this is the first we're discussing semi-structured qualitative interviews in this forum, so I hope everyone can deal with this. This is, a little bit, as an epidemiologist who first experienced this was outside of my wheelhouse, so I got used to it, so I hope everyone can get used to this. But basically, we identified individuals in 2 sites in Latin America, in Iquitos, Peru, and in Machala, Ecuador, who had dengue in the last 30 days, and we approached them, and most of the circulating dengue that they had was dengue 1. We approached them for basically a hour-long semi-structured interview where we first provided questions regarding specific symptoms, and then we allowed them to kind of spontaneously report additional symptoms. And then in those interviews, we also put a draft instrument in front of them and asked them kind of, did they understand it? Was it relevant to their experience? Trying to really get at, whether we were on the right track. So when we talk about kind of understanding dengue symptoms, this is the concept elicitation part of it. And I forgot to mention, so we identified 48 individuals, 20 adults aged 18 to 65, 20 adolescents or older children, 8 to 17 years of age, and then 8 caregivers of young children, 1 to 7 years of age in these settings. So when we asked them and tried to understand their symptom experience, not surprising, most frequently reported as well as most bothersome symptoms for the patients was fever and headache. They frequently reported, common impact to daily living, as well as physical functioning. Interestingly, there were a couple of newly identified symptoms that were spontaneously reported, so something like bad taste, which was interesting because we probed on loss of appetite, which was reported, but then bad taste was something that came up quite frequently. And I think it's important to note here that anyone who kind of experienced COVID, because this was kind of during the COVID period, we excluded from the interview process. So bad taste, back pain, dizziness, bleeding, and weak body were kind of identified, not asked about, but were reported quite frequently by the participants. And the figure on the right here kind of shows a breakdown of all the symptoms and signs that were reported, and their frequency by age group. The different colors represent the different age groups. So that kind of gives you a little bit of reference. And then when we talk about cognitive debriefing, which is where we put the instrument in front of the individual, it was very good to see that all the items that we included were understood and relevant, and we updated, the item as per kind of some questions from the participants. So early quantitative assessment. So this was, again, an opportunity that was presented to us by the folks here at SUNY Upstate. So this was there was a human challenge model study being conducted in 9 participants with dengue 3. It was a dengue 3 challenge. So there was an opportunity to put a draft instrument of the tool into this dengue human challenge model. Participants were asked to fill out the instrument for 28 days as per kind of the protocol for this human challenge model. Clinical measures and measures of RN anemia were also collected routinely. So this early quantitative assessment allowed, at least in an experimental model, to understand, will the form be completed, what's the completion rate, and also try and understand if it's actually tracking with clinical measures as well as, laboratory measures of dengue. So the peak symptom reporting on the DenVDD was between days 6 and 13, and that's what the figure at the bottom kind of shows, the various symptoms, symptom groups that were captured over time, and the average duration of symptoms was about 12 days. There was a bit of a tail on the right. You can see that dashed purple line. That was skin symptoms that tended to lag, and that actually carries forward into the psychometric validation. On the right is the comparison of the DenVDD item rating for fever and rash compared to clinical toxicity rating as per the protocol for the human challenge model. And you can see there's 2 different patterns, if you will, in terms of the relationship of the DenVDD item rating for fever versus clinical toxicity for fever, where there's maybe higher rating by the individual versus the clinician in terms of the fever experience. And there's a little bit of a converse with rash, where the clinician rating toxicity tends to be a little bit, more or higher as compared to the patient's own perception, if you will, of their rash experience. So psychometric validation, this is kind of a formal evaluation of the tool. So the objective was to evaluate the psychometric properties of the draft DenVDD as an assessment of symptoms of dengue illness in ideally children, adults, and adolescents and adults, as well as their caregivers. As I mentioned, we focused on the PRO version, which was really for adolescents and adults. This was done in a prospective cohort study in 2 sites in Asia, in Thailand and Singapore. So the study population were non-hospitalized dengue-positive febrile patients, as well as a subset of febrile patients who were dengue negative. And then the data collection was over 28 consecutive days, and during that period, additional measures of general health, so global measures of health, were also assessed as termed anchor measures to compare against the DenVDD. So there were 3 steps to kind of assessing the psychometric properties. There was the finalization of the instrument, there was assessment of its reliability and validity, and then it was interpretation of a scoring methodology. So we have this psychometric validation identified 81 dengue-positive participants, 56 that were 18 to 65 years of age, 25 8 to 17. Most of the infections were dengue 2 and dengue 1. So the first step of the psychometric validation was instrument finalization. So we assessed in this component ease and quality of completion, the distribution of responses for each item to make sure that we captured their relevance, and then dimensionality. So trying to maybe kind of condense or try and basically reduce down the number of items in the tool, and maybe potentially help group those items into domains. So the results were that the tool was generally very easy to complete, over 80% completion of the instrument. For most days, 96% of the participants reported it was very easy to complete. All the item options were endorsed. All the options, for each item were endorsed, meaning that there weren't any items, item scoring or options that the patient viewed as not being relevant to their experience. And then using confirmatory factor analysis, we saw that total score across all the items had the highest factor loading. There were domains identified with lower factor loading for fever, pain, GI, and skin. So that represents items grouped into those different groups. And then there were several items that were removed because of redundancy. And so the final measure, which I showed earlier, had 21 symptom items, and there were 4 domains. There was fever, pain, GI, and skin. And then there were 6 items that were kind of standalone that didn't actually load onto any of those domains. And you can see the list here. So for scoring methodology, this is just the next step. So you can see the instrument on the left with the different colors represent the different domains. Total is all of them, not the ones that are just in white. And so we had, as you can see, there were, apart from total, there were 4 different domains and the 6 standalone. And the scoring range was really a simple sum, right? Within each domain, it was basically the sum with not at all being 0 of each response. So a little was 1, the middle was 2 and 3. So for example, for the total scoring, you get 21 items over three potential outcomes. So you had a score range of 0 to 63. Figures on the right show the basically the trajectories of a few select domains. So the total mean score, fever and skin domain score, those means over time for patients. And you can see that very similar trajectories to what was seen in the human challenge, although because they were individuals presenting at a clinic with fever, the score starts higher and then goes down. Okay, and then step 2 was reliability and validity. So there were several measures that were used to assess this. So internal consistency reliability, so that's how well items within a given domain kind of represent the same concepts, and there was a strong correlation, particularly with total and pain domains, that the items within that were strongly correlated and seemed to represent the same concept. Retest reliability, so between days 1 and 7, and understanding that dengue illness is a moving target, this is kind of chosen as kind of the best time interval. To show consistency of reporting, and so there was a strong correlation of reporting between days 1 and 7 for the total and pain domains, moderate correlation for GI, and weak correlation with fever and skin, which could very easily represent the change in an individual's progression of their illness over time, so not necessarily a function of the tool, but really a function of the disease itself. Convergent validity with other measures of similar concepts. So as I mentioned, there were external measures that were administered concurrently over the 28-day period. And so those generally showed, for the total in each domain, showed moderate correlation. So the DENVDD at specific time points correlated with these more global non-specific measures of health to a moderate degree. And then we looked at differences in the domains and the total scoring between various known groups, so sex, different age groups, as well as dengue infection status. And we did see that there were significant differences in the mean scores for total and skin domain by dengue infection status. And I unfortunately don't have time to show that data, but I can show it if you're curious. And then the ability to detect change. So the idea here is that we wanted to make sure that the tool was able to be responsive and not incur ceiling or floor effects in terms of the intensity rating over time. And we did show that, as compared to external measures, it did show responsiveness to change. And then the last one is score interpretation, which I would say is probably still somewhat under development, but I think this is a good start. So the mean score and domain-specific scores were stratified by the global measure of illness. So if you remember, within the tool, there's a global measure of illness intensity. We compared that to the mean scores of all the domains, total and domain. And what you have here is you have the total median symptom score from days 1 to day 7 grouped by illness intensity items, so that global measure. And you can see that there's separation across the intensity scoring from good to very bad, which represents, I think, a good degree of internal consistency. Okay, just to summarize in the last 40 seconds, The DenVDD tool, designed with input from patients and scientific leaders and validated in an observational research setting, contains a comprehensive list of symptoms as well as general measures of health and impact that can be captured on a daily basis, demonstrates good ability to capture dengue fever symptoms when compared to clinical measures and external measures, and patients report ease of completion and relevance to their dengue experience. The mode of administration allows for ease of implementation in clinical and real-world research settings, and as I mentioned in the beginning, may help complement burden of disease estimations as well as help as a potential endpoint for assessing impact of interventions. And then with that, just want to say that it's intended for the characterization of the frequency and intensity of dengue symptoms and impact. This tool will be publicly available and publicly licensable, and it is developed with keeping the FDA and health authorities in mind as we think about potential use in interventional studies. And as I mentioned, we still have work to do for the observational work with the caregivers. So with that, I'm going to stop and thank everybody who was involved in this. Thank you.
Thank you, Morgan. Very nice work. And